Hyperpigmentation is a key feature of photoaging, along with the development of wrinkles, loss of skin elasticity, and facial volume. Pigmentation isn't just a cosmetic concern, it can be a sign of underlying inflammation. Post-inflammatory hyperpigmentation, for instance, arises from conditions such as eczema or other forms of dermatitis, all of which are part of what is widely referred to as sensitive or reactive skin. Acne scars are also a form of hyperpigmentation, beginning with post-inflammatory erythema or redness immediately after acne resolves. This redness is a sign of inflammation, which later becomes pigmentation as the melanocytes begin to produce melanin. Melanin deposited in the superficial and deeper layers become what we know as hyperpigmentation. Traditionally prescribed tyrosinase inhibitors such as hydroquinone reverse these processes by blocking the melanin synthesis pathway. However, hydroquinone is also associated with skin irritation, which can occur even with short-term use. Sensitive skin, for example, does not respond well to hydroquinone therapy. It is possible for flare-ups to occur, necessitating the use of topical steroid therapy. For instance, the 3-in-1 hydroquinone-retinoid-steroid formula, known by its trade name Triluma, was specifically created to counteract these side effects during the treatment of melasma. Such an approach isn't a solution either. Those with sensitive skin are susceptible to side effects or irritation caused by barrier disruption. Is it possible to treat hyperpigmentation without the use of prescription topicals? Hyperpigmentation treatment in sensitive skin types deserves a more in-depth study. Join me as I dive right into the science of this conundrum. I'll share the latest research on bacterial ferment filtrates as melanogenesis inhibitors, alternative tyrosinase inhibitors, and the exciting field of functional dermatology. Sensitive skin is a state of hyperreactivity, and key to understanding this is the concept of barrier function. We already know about the brick wall model of skin, which means that once the skin barrier is damaged, we know also that the proteins associated with the structure of this wall also changes. Ultimately, this is what stimulates inflammation, leading to tissue damage, further exacerbated by what we call the itch-scratch cycle, which is the role that neurological factors play when it comes to sensitive skin. In dermatology research, there exists a sensitive skin model, which is essentially one that mimics actual skin barrier damage. Researchers proceeded to investigate the role of cosmeceuticals as well as its effects on the damaged skin barrier. The following are a list of extracts that have been identified for its efficacy. We have oat extract, olive leaf extract, brown algae, stachykos, and erythritol. These extracts are chosen for their ability to scavenge free radicals, inhibit hyaluronidase, the enzyme responsible for the breakdown of hyaluronic acid, an important structural protein in the dermis. I want to now draw your attention to what I call functional filtrates. These refer to bacterial ferments such as those from galactomyces and lactobacillus. The story begins like this. Elderly sake brewers were observed to have wrinkle-free, youthful-looking skin on their hands, which were in constant contact with the sake fermentation process. And this was in contrast to their faces, which were wrinkled and filled with pigmented spots. Clinical studies done by scientists then demonstrated that galactomyces and lactobacillus ferment filtrates were able to improve fluctuations in redness, roughness, and pore size. They were also involved in alleviating mask-induced skin irritation in the era of mask wearing during the COVID-19 pandemic. Now, how exactly do bacterial ferment filtrates work? First of all, they upregulate ceramide production via filaggrin gene expression, which is what we know as the key player in healthy skin barrier function. They are also what we consider tapinarof or tama-like. These have been used as alternative therapeutics for inflammatory skin diseases in dermatology like psoriasis and eczema. Ferment filtrates are nature's very own tamas. Beyond that, bacterial ferments have an inherent antioxidant capacity. This additional antioxidant effect is what neutralizes damaging free radicals, blocking key mechanisms in the process of inflammation, for instance, cell senescence, which is the process of cells falling asleep. By doing so, we are reducing stress on the skin and enhancing skin cell repair. Cosmeceuticals containing bacterial ferments also demonstrate increased caspase-14 enzyme expression, which are essentially enzymes that are activated by the phytochemicals present in plant extracts. There is also increased expression of tight junction molecules, which affect the connections between the cells that make up the skin barrier. And this ultimately affects the permeability of the barrier and helps to restore healthy skin function. Key to bridging the gap between the needs of hyperpigmentation sufferers and those with coexisting sensitive or reactive skin is understanding cosmetic formulations that are based on the latest research in both skin barrier function as well as functional dermatology actives that target hyperpigmentation. Ingredients tested on damaged skin models in the laboratory setting can be of significant use to those seeking alternative tyrosinase inhibitors. We have discussed in the short tutorial botanicals and phytochemical extracts as well as bacterial ferment products that target multiple pathways of skin inflammation and reactivity. In our case study of an ideal cosmeceutical regimen for those with eczema, dry sensitive skin, and coexisting hyperpigmentation, we have distilled that the most important foundation of such a product must include ceramide and natural moisturizing factor dominant skin care. Being retinol and retinoid-free also minimizes the risk of skin irritation. The exciting world of functional dermatology means that there are now alternative tyrosinase inhibitors and novel melanogenesis blockers that can play a key role in the future of cosmeceuticals.